Welcome back, everyone. Hope you had a good break. My name is Mike Iacona. I'm the finance director here at the Rich Earth Institute, and I'll be moderating this next section. So this is the fertilizing with urine section, and the first presenters are doing a presentation titled Fertilizing with Human Waste for 25 Years, the Many Benefits. This is presented by Hilda Maingay and Earle Barnhart. Hilda Maingay and Earle Barnhart are directors of the nonprofit Green Center. They have used the urine and compost from their urine diverting compost toilet for 10 years in their vegetable gardens. They advocate for widespread use of urine diversion on Cape Cod in Massachusetts, and they are developing a waterless in-wall urinal called the P-Pod. Just a reminder, we're going to do 15-minute presentations in this section with 5-minute Q&A afterwards. So you can put your questions in the chat and we will address them after each presenter. So Hilda and Earle, go ahead.
This one. No, this one. No, this one, slideshow.
Yeah.
That one. Good.
Can you see it? Are we on?
Yes, you're on. Go ahead.
Good. Great. Thank you for having us. So fertilizing with human waste nutrients keeps the nutrients out of the waste stream. So we have a big pollution issue on the Cape. The way my screen is, it covers up a lot of stuff, all our viewers. So I don't know if that can change. Anyway, we'll let you know. We'll just keep going because a lot of it is now covered up for us. We are going to show you first how we divert all our human wastes. We have a separate villa and a QB. Separate villa is a Swedish toilet, a urine diverting toilet, all is waterless, and it goes to the full circle composter that Abe developed. And we have a little QB that Rich Earth has developed. We have a P-Pod, urinal in the wall, and a urine storage in the basement. Everything is waterless. So here are the components. And the cost for us is the installation has been $7,000. The maintenance over 10 years has been $75. The electricity that we use for all the systems in the house is for 10 years has been $400, $40 a year. Doesn't matter how many people are in the house. We have 2 apartments and we have 3 bins that are resting all on the same fan. We have actually solar energy, so it doesn't cost us anything. So the total cost has been $7,600 over 10 years. It will be less because we don't have investments anymore. We store it outside. We have a little drum as well as a big IBC toad. That's really what we're using now, but I just wanted to show you how we transfer the urine from a QB into a barrel, either the IBC toad or a barrel. And that means that there is absolutely no exposure to people. This method has been approved by the Farmers Board of Health. And a lot of people use this very simple method. And there's a company, Wasted, that picks up the urine if people don't want to use it on their own land. So how much water did we actually use? Well, if we don't flush, we save about 4,000 gallons per year per person to not flush the urine. If we also not flush the feces, in our case, that's the case, we will kind of save 6,000 gallons. So over time, over those 10 years, we have saved about 120,000 gallons of water, and it's approximately $1,300. All depends on what the town does and how they charge for things. And this part is very important in the next slide. We want to see how independent we can become from all our big organizations that provide us with the needs, such as water, such as sewers. We want to see how independent we can become. So we had to figure out how much water we use. And we have done that recently. and we go into that more detail later, but we found out that over, we've done it for 6 weeks now, we use an average of 25 gallons per day, which is much less than we thought we did. We couldn't really figure it out because we use most of our water for our agriculture. But that was really important to know if we ever want to have housing that becomes more independent from expensive infrastructure. We have a cistern already that holds 1,500 gallons. We collect the water from the roof, and eventually we'll connect that to our house. And now we know that our system holds about 2 months' supply for our household. Now, how much fertilizer do we actually produce, and how much land can it actually fertilize? So we did a pilot project together with Mestec. Brian is here from Mestec on this summit. And we found out that our household of 2 adults, home basically full-time, because our office is also at home, produced about 15.9, say 16 pounds of nitrogen per year and 1.9 pounds of phosphorus. Both of those are very important for our ecosystems because the phosphorus pollutes the freshwater more and the nitrogen, the saltwater bodies. So that report is available for people if they want to see what we did, how we did it. We had 41 households participating in this program.
Cubies, yeah.
And they only use cubies. So this is a view about our gardens. You can see our house with the solar greenhouse on the front. We have about 9,000 square feet of gardens between those 3 gardens that are on the arrows, and they are exactly the amount that we can use all our fertilizers, because we have about 16 pounds of fertilizer of nitrogen per year that we produce, and the recommended application rate is exactly right for the amount of land that we have. Our soil has gotten so good, though, that I think we have to start using less so we can start using some of this for lawns and shrubs and trees in the next few years. So how do we apply it? Well, it's kind of the opposite of what you just saw in the movie. We do it all by hand. We are very careful about not wasting any of the nitrogen, either into the groundwater or volatile. So we make sure that we dilute it if we put it around plant, and make sure that we water it right afterwards, or that the soil is very moist. If the bed is new, on the left side, you see Earle pouring it straight onto the bed. And then right after it is applied, then we put water on it to make sure it goes into the ground. You see that the IBC tote on the left side. And on the right side, we just covered up the feces. We composted for 2 years, which is what we have been told to do to make it safe for the food production. So now we have other issues here that we would like to show. We try to avoid greenhouse gases as much as possible. So all our agriculture is done without machines, without fossil fuels, no fertilizers from commercial fertilizers. Any food waste that we have all goes back in the garden. We dig a trench, put it in the garden, and cover it up so that we don't lose any nitrogen. And all the landscape debris, and we avoid to have anything go to landfills. All the nutrients that we buy, either as fish food or chicken food or animal food or people food, all the nutrients get recycled back into our landscape. And this is how good our soil has become. It was from really, really poor soil on low on organic matter. It's now a 1.5 foot deep of good soil, and all the soil tests came back really high. We have no salinity whatsoever. So just give an idea of how much production we have. This is what we produced this year. Just a quick overview, just to show the value of our waste that we use to grow our food. And we have grown basically all our vegetables. And last year when I calculated, we also grew all our energy, our carbon basically. And this is an overview of what it looks like in our house. We dry, we eat fresh, we store in our basement, and it provides our food for the rest of the season.
So this talk is also meant to show the benefits of recycling your nutrients locally on the larger society. So one of the benefits on Cape Cod is we do not have to import food to the Cape. 460 million pounds of food are trucked onto the Cape every year. All those nutrients go into the groundwater. 6 million pounds of fertilizer are trucked onto the Cape every year for lawns. That goes into the groundwater. This can be avoided. What happens on the Cape is these nutrients go into the groundwater, they go through freshwater ponds, they pollute the ponds, make the toxic algae. It produces wastewater on the coastal estuaries, pollutes the water, kills the fish. It pollutes all the groundwater in the entire Cape as waste nutrients and waste fertilizer nutrients. The Cape imports 6 million pounds of fertilizer a year, which is made with fossil fuels at some remote place, or it's made out of recycled sewage sludge, which contains microplastics and PFASs and other contaminants. You can avoid importing and using that kind of fertilizer if you use local recycled human fertilizer. Synthetic fertilizers produce huge amounts of CO2 in their production. You can avoid that by not having to make that, to replace it every year. Human waste fertilizer reduces costs of wastewater systems. On Cape Cod, there are some towns that have sewers. The sewers are extremely expensive, hundreds of millions of dollars for small towns to build them and maintain them. The state has also required now that people with septic tanks, of which 85% of the people on the Cape have septic tanks, have to make an additional special septic tank next to the first one, which makes some of the nitrogen goes off as gas, but doesn't remove any other nutrients. It all still goes into the groundwater, except for some of the nitrogen. The homeowner has to pay for this new septic system, which costs $35,000 to $80,000 each.
Plus maintenance.
Plus maintenance every year. You can avoid these expenses if you do urine diversion and nutrient recycling of human wastes. People know this, 80% of the nitrogen and half the phosphorus is in the urine, and the urine's only 1% of the volume. You have to deal with that 1%. So how can diverted urine replace the costs of the state requirements? On Cape Cod, the state requires that for each of our coastal estuaries, a certain amount of nitrogen has to be removed by law. And they're trying to do this with sewers and with these special septic systems. And they're requiring in one particular estuary, which is the next one in our town, which is soon to be sewered for hundreds of millions of dollars, that a certain amount be removed by sewers or septic systems. We did calculations, and we've discovered that if homes in that watershed, if half the homes in that watershed would use urine diverting fixtures and divert the urine and get it out of the watershed, the town would not have to build these sewers. You would meet the goals, and you wouldn't have to have any of these expensive industrial infrastructures built instead. You could divert by urine diversion in these watersheds a huge amount of value of fertilizers, which are now imported. And it's possible you could recycle the nutrients from the local wastes and use that as fertilizer for lawns and replace these various imported fertilizers. This is what a special septic tank looks like that is put in for homes. And the goal here is by the state is to reduce the concentration of nitrogen coming out of the septic tank. They are not interested in the amount of pounds coming out, which is what the law says has to be reduced. They are focusing intensely on the concentration of the nitrogen coming out. the concentration is meaningless, because if it's the amount of nitrogen coming out that counts. If you use twice as much water, the concentration goes down. If you use half as much water, the concentration goes up. But the amount of nitrogen coming out is exactly the same, and they lose track of this idea. So we did experiments in our house. We've captured the gray water coming out of our house. Our house diverts all the human waste nutrients by various fixtures. And we put a little sump pump into the container that comes out of our septic tank, and we've measured the volume of water coming out of our house. No one has done that on the Cape, essentially. And we used the amount of water coming out. We had Maztec test the concentration of nitrogen in the water. We multiplied the two together, and we discovered that our nitrogen coming out of our house is 7/10 of a pound. About 16 goes in, 7/10 comes out. This is lower than these special $80,000 IA septic systems put out in their effluent. So we think people should be able to do urine diversion and waste reduction by eco toilets instead of these expensive special septic tanks.
And it's also very reliable. It's always the same. And all these fancy systems are not always reliable. And there are good reports on that from one particular project that's called the Schubel Pond project.
So the normal solution here is to build a septic tank or sewer, or put in special septic tanks. These are expensive. It's calculated that to remove a 1 pound of nitrogen by using a sewer costs $300 to $400 per pound, and the average person makes 8 pounds. So the average person is paying $2,400 a year to have it taken out by a sewer. Same with these special septic systems. Urine diversion can cost about 1/10 as much as a sewer or a septic system. So we're lucky, actually. We have 85% of the people on septics still and not on sewers. We haven't built sewers, so we might be able to avoid these sewers, which may cost several $1 billion on Cape Cod.
It's more an analog of the lesser developed countries that never had phone lines, and they avoided them all. They went straight to cell phones. And we still have both systems. So we want to be the same thing. We just want to avoid these big systems and go straight to the more ecological, low-tech systems.
So on a global level, the recycling and recovery of nutrients on a home scale does things that benefit the globe. It reduces greenhouse gases on the globe. It conserves potable water wherever it's done. It reduces water pollution and ocean pollution, where all the nitrogen and phosphorus and other minerals now end up. It conserves non-renewable resources, such as water and phosphorus. It increases food security wherever it's done, over the entire world. You don't have to transport food thousands of miles across the globe from place to place. And it increases economic and social equity. People all have the same amount of nutrients that they produce every day, and they can produce their own food in their own location, and it becomes equitable.
So the benefits of human waste fertilizer is the food security, of course, if you have land. The water is cleaner, and the air is cleaner, and the communities will become more affordable. And affordability is a big issue on the Cape, but also in many other places in this country. And this is our strategy. The strategies we research emphasize a minimal reliance on fossil fuels and operate on a scale accessible to individuals, families, and small groups on behalf of the planet. And everything we do individually affects the whole. And that has to be kind of instilled in people, that it's not like, oh, well, the government is going to do it, or someone else is going to be responsible. We are all responsible, and we all affect the whole. And this is what we must consider. It's the next generations that we're doing this for. We have had our lives, but this is why we're working really hard to make our planet a little bit better for these kids. Thank you so much, and please be the change.
Thank you, Hilda and Earle. Thank you for sharing that presentation. It's inspiring to see your commitment to diverting and recycling your nutrients. So we could probably take one question. I'll just take the first one that came in. It was from Lonnie Goldthorpe. In addition to putting your human waste in trenches, do you also compost?
Yes, we do compost, but we don't compost human waste. We only compost any materials that is too big to compost directly in the garden, which we didn't go over, but we compost in the pathways. But we have big compost piles for things that are too big, branches and stuff. And we don't use any food in there, and we don't put any human waste in there.
Okay, thank you. And there are a few more questions in the chat. Maybe you could answer them directly
Sure.
in the chat. That would be great. Thank you.
Mm.
Okay, so we're going to move on to our next presenter. The presentation is titled From Yellow to Blue, Exploring Indigo Cultivation and Fermentation with Liquid Gold. This is presented by Rosi Olivan. And Rosi is a science teacher at the Putney School and a fiber artist at itequiya Lab. She is dedicated to learning and teaching sustainable dye practices. And a bonus fun fact, Rosi has served as a volunteer and a board member at Rich Earth. So Rosi, go ahead.
Hello. I think I can share my slides now. And just triple check that everybody can hear me.
Yeah, we can hear you. Go ahead, loud and clear.
Awesome. All right. Let me just put this aside. Thank you all so much for being here. I'll just share with you some results from a project that is really important and meaningful to me that involves a little bit of science and art. So I've been growing indigo for 4 years, and maybe for those of you that don't know what indigo is, it's an organic pigment. And you can extract it from different plants, but you can also synthesize it chemically. And I'm just getting distracted by, I don't know if everybody can hear the presentation, but I'll go. And I was just saying that I have been doing this for like 4 years. The first year I just grew a little bit of some plants at home, but did a lot of experimentation. And since then I've been like teaching some workshops and trying to scale up this project. So here I am to talk about a pilot program that we run this summer. We grew Persicaria tinctoria, a hybrid. I think it's Sanbon and Jehimiba for anybody that is interested. And we grew these plants in 4 different sites at the Putney School, at the Richard Institute, and with 2 dear friends. The participants, following the protocols that the amazing team of the Richard and myself like put together, and participants could choose control with no urine, low dose and high dose, following this amounts per season, and we did 2 applications per season. And I'm gonna show you some of the results, and most of this is also like me trying to figure out how to measure the impact of indigo, of the urine on indigo plants. So we measure the height of the plants, and these are some of the sites. It looks like the high dose maybe it was a little bit lower than the other ones. We also measure the mass, and this is interesting because even if the plants were like shorter, they cannot produce the same amount of mass in total. And we also kept track of other things such as color. And I chose this particular picture because I think you can see that the control group was like yellowish in comparison with the low and the high dose on this one. And I think Tatiana has a bunch of notes also about like how the plant was more resilient to drought during the season, and we just kept track of other colors. In addition of this, we also play with a little bit of art. You can extract the pigment from the leaves. So you can use this pigment with the fresh leaves, or you can do an extraction to get the pigment. So we did two methods for the fresh application. One is called the rub salt method, where you get some leaves, add a little bit of salt, and you like kind of smash them to try to force the dye molecules to stay on the fabric. And I think last time I said that I think I'm biased about like which one performed better. And we will need like colorimeter to really get to measure the colors that we extracted. We also did another technique that is popular with some dyers. It's called tataki some. You hapasome, I think for other people call it. You take the leaf, put it on the fabric, and like hammer it. So you're by pressure, you're hoping that the dye molecules will stay on the fabric. And this is a close-up of one of them. This is really fun. So I think we were trying to measure things, but also have fun with this project. And the younger leaves, I don't know if you can notice, but the younger leaves tend to produce more pigment. All right, so before I continue, I'd like to take you on the tour of the molecule, like how it changes from plant to pigment and eventually to dye some items, just so you understand my goal of what am I measuring. So when you have the plant, imagine that you have like a molecule like, let's say that it looks like your hand connected to a sugar molecule, like a glucose. And then this molecule, we hopefully try to break it down in an aqueous extraction. So you get one, the molecule, and then I'm sorry if you're hearing some noise, I'm at school and there's a transition right now. So I apologize for that. So you have the indoxyl molecule, and you're hoping to find another molecule similar to this one to make the blue pigment, the indigo tin. And this one can be used artistically in some applications but for some like dyers, like if you want to dye fabric, you still need to reduce this molecule to make it soluble so you can fix it on the fabric and eventually it gets insoluble. So it's like this beautiful dance. All right, thank you for that. So I'm sharing this because initially I think I was pursuing the path of how much indigo tin can I get from the plants. And I realized that this was really tricky because even from from the same batch, I could have different colors because there are two steps that I need to do. So I was like, maybe this is not the right way to do it. I also I want to thank Sky McAllister, an expert on indigo, and she was helping me to realize that maybe like going after this was also like a colonialist approach of trying to go for like the darkest color and not using what I have. But still, I think I'm gonna measure with my students in the spring the purity if we have the equipment. And then something else that I noticed was the indoxyl levels. I'm gonna play this video for you all. So this is the control, this is the low dose, and this is the high dose. I'm gonna play it again so you can see the difference. So I kind of knew that maybe this could be the way in which I can prove that using more urine, it was gonna lead to more pigment. And at the time, I didn't measure exactly the mass and the water that I used for the extraction, so I knew that I had to do it again. Then we went to our second harvest and with new observations. This time, at least for my side, it was really interesting how the control group was performing lower and the other two that with the urine were higher. I think it's because we allowed the plant to have more time to grow. And something else that I noticed was that how like spread the plants were to the sides was like more on the high dose than the low dose and the control. These are the measurements. So I think we're still figuring out what are the parameters that we need to measure for this project. Something else that I noticed was that there were more growths on the plants that were fertilized with urine rather than the control one. There were like some for the control, like smaller ones. So I think we're gonna try to measure this next year. And then going back to the indoxil, which is the aqueous extraction that we do, I had to run another experiment. This time I measure the mass, the water, the temperature, everything was just control. And after 36 hours, I could see like some indications that these two were producing more. This is a control, low and high dose. This was after 60 hours. So for me, this is indicator that I need to pursue the measurement of this solution. However, I think the best way to do it is by measuring the absorbance of each of them. And I did try using a regular visible spectrometer, but I need to use a UV-vis spectrometer because the indoxil reacts to UV light. And so that will be next time. Then this is how like with visible light will look each of the extractions, the control, low and high dose. And I extracted the pigment and these are the results that I got. I used the same procedure for each of them and I got more pigment for the high dose. I still need to compare the purity, but that's again something that I'll do later in the spring with my students. I think this for me was I don't know, like a lead that I need to continue with this and try to, I don't know, continue with this experimentation. Then, so what's next? And this is one story that I'm sharing with you. I think in the spring, I'm gonna test the purity of these pigments. We're also gonna, we have some dry leaves and we're gonna try to extract the pigment with a different technique. For the next season, I think we're gonna add a new study group. I know that many of the dyers, they fertilize the plant every other week. So that might be interesting to see how it reacts. Keep track of the new growths. And I think I really wanna measure the indoxil levels using a UV-vis spectrometer. So that was like one story. I think I still have time to share the other one. So now urine can be used not only to fertilize the plants, but it can be also used in the process of reducing the indigo to be used on textiles. And for that, I'm going to introduce a concept that I think many of us are familiar with, which is like the triangle of fire. That in order to have fire, you need to have like the oxygen, the fuel, and the spark. So I call for the indigo, like the triangle of indigo. And again, this is for the pigment to being able to dye fabric. So traditionally, you need three sources. You need the indicant source, which is the pigment, a reducer, and you need an alkaline solution. The beauty of using the urine to dye fabric, which is called sick fat, by the way, is that you only need two ingredients. There's like a third one, but it's like kind of in there, right? So you need, again, your source of indigo. You need the alkaline environment, which is provided by the urine, but you also need, like, you have the perfect environment so the bacteria that you can either get from whatever is around you or already on the urine can reduce the molecule so you can use it. And I think for me, it was like, what? This is amazing. And honestly, before I mention the other thing that I'm going to say, I've been trying so many different types of regular vats, and I couldn't believe how easy it is to use the sick vat with urine. And I'm so amazed by the results, and I'll share a few sums of those in a second. A couple other things that I want to share before I move on is that I think this technique was really popular in Europe. I don't know, 1800. But I also know that in Mexico, urine was one of the components that you use for like an indigo vat. Other advantages of this? Well, you can use the leftovers on your bed whenever you're done or you're not getting more pigment to fertilize your plants or your garden, and you're saving water. So I think this is for me a lead that I need to keep pursuing this. So I'm going to show you some of the results that I got. This first slide that I'm going to show you was using commercial indigo that I bought. I think this one was from India. The other one that I'm going to show is from Turkey. So I started with a very small bed, 1 gallon, have it outside under summer. And the colors that I was getting were like amazing. I was like blown away by how easy it was to do this. Then I move on and did like a 5-gallon vat. And I've been doing like so many projects. I wish I could put all the pictures here. And the cool thing is that in the last, the summit in Cape Cod, someone provided me with this piece. So now I did a collaboration with Dirt Wolf Dyes, the artist. So this piece is gonna be donated to the Research Institute. So I don't know, I think this project keeps growing and I'm really happy to see this. And the colors are darker because I'm getting to know my vat, and now I think it's happier, so I can get like darker colors. And this is in cotton. The other ones are protein fibers. And I think this is my special moment when I was like, maybe some of you are gonna ask, have you used a pigment, right, that you extracted and then reduce it with indigo? And the answer is yes. I only used 25 grams of one of the pigments that I got. I would make like a 1 liter vat, which I think is the smallest that I've ever made. And I got beautiful blues on local wool. I've been just testing other things. This is, I think, 2 deeps that you put like the fiber twice, and this one I think it was like 3, and it's really, really dark. And the scarves that I'm wearing are also dyed with this indigo, local indigo, and reduced with urine. This is 2 dips on the bed, and this is 4, which it leads to, I don't know if you can see like darker colors between these 2. Plus another surprise that I guess is another color that you can get from indigo as well. So I guess I'll take questions. I'll just want to let you know that I'm going to keep experimenting because this project is really, I love indigo. I think indigo could be a lifestyle or an obsession sometimes. I just want to keep having fun, building community, making art. And again, this plant is so special because you can not only get like blue, you can get like yellow. These are 3 different inks for a project that I did with all local indigo. You can get a yellow from the leaves, and there's another molecule called indirubin that you can also, it's an isomer from the indigo tin molecule, and I know it's just fascinating. And I just want to say thank you all, and thank you to the Research Institute for I don't know, listening to my ideas and working on this together. And I'm hoping that we can find some grants to do this again next year. And this is the email for my job. And this is, you can find me on Instagram on itequiya love, where I think is my playground. And I think that's pretty much from me. So I guess I'll take any questions if there are any.
Thank you, Rosie. Yeah, that's wonderful to see that. And yeah, just really awesome stuff you're doing. I appreciate the combination of science and art and working with kids. I'm excited about it.
Yeah.
Let's see. So I don't see any questions in the chat at the moment. Probably have time for maybe one question if anybody wants to ask a live question. Oh, here's a question coming. How do you test the purity of the dye?
With an spectrometer, you need to dissolve the dye on a specific solvent. I still need to figure that out. And then you run specific wavelengths, and you can measure how much, compare it with like 100% sample, how much of your purity gets. I can send more details directly to the person. but
Great.
it's
Yeah.
using spectroscopy. Mhm.
Okay. Yeah, maybe you can send that info to Brian or to the group in the chat. That'd be great.
Yeah. And I think the last thing that I would say is that someone told me that in order to really do this, you need to measure like the percent of the pigment on the leaves, which is like HPL, like other things that are really cool.
Okay, good to know.
Mhm.
Okay, so next presentation is Krisztina Mosdossy, and she is presenting a presentation called Composting at the Municipal and Household Scale with Urine and Biochar Increases N2O Production and Impacts Nematode Bioindicators. Dr. Krisztina Mosdossy is a soil ecologist, longtime peace cycler, and postdoctoral associate at Cornell University. Her research focuses on the carbon and nutrient cycling of organic materials by nematodes, fungi, and microarthropods. Chris will be teaching regenerative agriculture at Evergreen State College starting in the winter of 2026. So Krisztina, go ahead.
Thank you so much. Can you hear me and see my screen?
Yeah, we can hear you and see your screen, so you're good to go.
Amazing. Thank you very much. So excited to be here and for the second time today. And I love the Rich Earth Institute, so thanks for hosting. So I like to start by thanking the people that I work with, because without them, I would not be able to do any of the work that I'm presenting to you today. So huge gratitude or Watekinamuraton to the Nelson Lab, to the Dillman Hill Student Organic Farm, as well as various support members of the Cornell University and Dairy One, where I sent some samples for analysis. And of course, I did this work on Haudenosaunee land, and the Haudenosaunee Confederacy founded the first participatory democracy of the Ghanaian-Aragoa, the Great Law of Peace. So I want to acknowledge that without that, well, we wouldn't have the democracy system that we have today, and I wouldn't be able to do this work because I wouldn't have had access to that land. Okay, so we all know that urine is a very good fertilizer, and we're all on board with using it, as we've seen many examples today, yesterday, and we will again tomorrow. However, in some cases, urine is considered taboo, where it's not as culturally accepted. Putting it directly on crops is not so desirable. However, in those cases, sometimes composting with the urine is more accepted because it's more indirect. So you can create a fertilizer with the urine and then use that compost for growing crops downstream. However, there's not too much information on how urine does as a compost amendment and what the potential benefits or downsides are. For example, maybe if you're adding urine to a carbon source, you could be creating nitrous oxide, which is a potent greenhouse gas. So this work was exploring those ideas. So I did this using super sacks, their cubic meter scale, kind of mimicking a municipal scale or backyard scale composting, using what the Nelson Lab calls hours or organic underutilized resources. And in this case, that was agricultural residues, so maize stover or corn stalks or wheat straw. And these are taken from an agricultural field. And we considered leaves that we collected on Cornell campus to represent yard waste. So this is kind of the two different scales and systems that we saw this potential composting method being applicable to. And the leaves were a big mix. It was anything that was collected on Cornell campus. So oaks, maples, larch, locusts, etc. And we use nematodes as a bioindicator for compost maturity and quality. And just to, there's too much information on the slide, but just to give you an idea as to why we use nematodes, not only because that's my specialization, I'm a soil ecologist, and I've used nematodes in my work for many years now, but they're excellent in indicating nutrient availability as well as the microbial community and what they're doing, because nematodes are the main predators of bacteria and fungi are the main cyclers of nutrients like nitrogen in soil and compost. And then there's the different categories of nematodes that indicate whether your soil or your compost is in an early stage of decomposition or nutrient availability. So kind of if there's a lot of nutrients available for microbes to be eating, then that means there's a lot of microbes for nematodes to be eating. Nematodes are kind of like the hyenas or the lions of the Serengeti, where the bacteria and fungi are kind of like the gazelle and other antelopes of the Serengeti. So they have that kind of relationship, and nematodes are super abundant, so they help us understand what's going on with the nutrients. And then past work, Steele et al., for example, is a Steele et al. 2013 in Ecological Indicators journal, have shown that different communities of nematodes exist at different times through the thermophilic composting process. So, for example, looking at the earlier stages of composting, you'll find more bacterial feeding nematodes. And then towards the end of the composting process, you'll find more fungal feeding nematodes. And I'm not, unfortunately, going to be able to show you results of the communities today, but just to give you an idea of how nematodes have been used to understand the composting process in the past. So this experiment using super sacks was conducted on Cornell campus over 2 different years. So the first year we were looking at wheat straw and maize stover, as well as leaves. And we had 3 replicates of adding urine versus adding water. So any sack that didn't have urine had water added to it. And we had a total of 18 sacks. Whereas the second year, we just looked at leaves. And again, we used urine and water. We had 4 replicates of each fertilizer treatment. And we also included biochar and no biochar in this experiment. And we had a total of 16 sacks. And I'll just go through a few of the results that we've seen so far. I'm just going to move you all around so that I can see what's going on. So this is looking at temperature. So on the y-axis, we have temperature in Celsius. And then the various hours or the organic underutilized resources in each little box. So we have corn, which we also had a plus or minus biochar treatment, but there were fewer replicates there. And then leaves, wheat, and then a mix of wheat and corn. And we found that overall, sacks that had urine applied had higher temperature than sacks that had water applied. And this was especially true for, so you can see the asterisks is with leaves, wheat, and this wheat corn mixture. And there wasn't so much a difference with just corn stalks by themselves. And we didn't see any biochar effect. And then this is looking at the chemistry of the leaves, specifically from the second year. And we found overall that there was more, like a higher carbon to nitrogen ratio, more lignin, and then more of this lignin adjacent molecule in the water amended sacks versus the urine amended sacks, which gives me the indication, and these are just preliminary results, but gives me the indication that there was more decomposition happening with the urine applied sacks versus the water applied sacks, because there's more of those more complex molecules broken down. And again, we didn't see a biochar effect at this stage. And then we used a portable Lycor device to measure nitrous oxide in these sacks. So at first we didn't see any difference between treatments, and this is just for the leaves of the second year of the experiment, without biochar and with biochar. We saw no difference between the biochar treatments and no difference between urine versus water. And that's just in November of 2024. And then moving on to 2025, we saw gradually over time a difference with the urine applied sacs versus the water applied sacs. And again, no biochar effect. But when we did have biochar, urine was almost 20 times, was producing almost 20 times more nitrous oxide than water. And then urine with biochar versus urine without biochar was producing 6 times more nitrous oxide. So biochar is in the literature often seen as a way to mitigate nitrous oxide production. And in this case, we're actually seeing the opposite. But so far, this is still early days and early in the season. This is April of 2025. The scale on the y-axis, you can see, only goes up to 40 or 50 milligrams per square meter per second. So we're not producing very much nitrous oxide. This is relatively low amounts of nitrous oxide. But then it does increase as time goes on. So now the scale is up to 500 milligrams per meter square per second. And we're seeing this similar results where with biochar and with urine, we're producing more nitrous oxide. So that's urine with biochar compared to urine without biochar, we're producing more nitrous oxide, as well as producing more nitrous oxide with urine compared to water. So not the most positive results in this case, but bear with me. And then overall, as time moves on, we're seeing these effects even without biochar. So urine is producing more nitrous oxide than water with or without biochar. So taking all of those time points together, overall, we're seeing no biochar effect. So that's interesting. It kind of evens out over time, perhaps. And then this pretty obvious effect of urine producing more nitrous oxide. So how this related to nematodes, where we saw actually more nematodes, so higher abundance of nematodes, we counted them in the microscope, when we had biochar versus no biochar. So in cases where we're producing more nitrous oxide with biochar, we're also having more nematodes. And then we also saw more nematodes with water compared to urine. And what was really interesting with the nematodes is that we saw an abundance of what's called a dower larva. And these are a life stage of nematode communities that indicate low quality, low resource availability. So not a lot of microbes available for nematodes to eat. So they kind of exist in this dormant, non-feeding stage called a dower larva. And we're seeing an incredible number of these, 75,000, 90,000 nematodes. These are very unusual numbers of nematodes. You don't usually see this many in soil, for example. You'll see a few hundred up to a couple of thousand. So this is indicating to us, at least in these 2 sacs with urine and 5 sacs with water, so not across the board, but these specific sacs had these incredible numbers of nematodes. So that was kind of a red flag for us, suggesting, oh, perhaps these super sacs are actually producing a anaerobic condition that is not suitable to nematodes, and perhaps is not a very good way to compost in general, because it's a static sac. We're not aerating it, we're not moving it around. So overall, we saw that biochar increased nitrous oxide production and nematode abundance, and this was especially true with urine. So this could indicate that there was higher microbial biomass and activity with biochar, which makes sense. We know in the literature that biochar creates these nanopores where microbes tend to hang out, and offers an environment where they can thrive and consume more nutrients. So that part makes sense. And then perhaps these anaerobic conditions are leading to them producing nitrous oxide, or whatever conditions are creating this environment that leads them to produce nitrous oxide. And then with these very specific 7-ish sacs, we found these Dauer larvae, which indicate low quality compost. So that would be not compost that you would want to put on your garden. It's very high carbon to nitrogen ratio and likely low microbial biomass, which would also be unhelpful in your garden. And so we'll be able to know more about what's going on here when we have results from our DNA analysis that tells us about the nematode community composition. And like I was showing earlier, when you know the ratio of bacterial feeders to fungal feeders and how many predators are present, that tells you what stage you are in the thermophilic composting process and kind of how your composting process is going. And so I think overall, this is showing that we explored this way of using super sacks as a composting method that is potentially low maintenance, easy to do in your backyard, a way to compost these agricultural residues versus yard waste, and using urine as a way to increase the nitrogen content in the compost. But because we didn't aerate them, we saw these results where we were producing a lot of nitrous oxide, and the nematodes were indicating that these are potentially not very good living conditions for microbes, meaning that the compost is not very high quality. So we have started an initial pilot study on what these same conditions look like, or sorry, these same treatments look like in an aerated condition, which of course is more maintenance, it's more work. Many of us, I'm sure, have composted before, and so turning a compost pile is always very laborious and energy intensive. So that's why we wanted to try the static option first. But I think that will lead to very different results with and without biochar, with and without urine, and hopefully less nitrous oxide production. So yeah, with that, I will take any questions. Thank you for listening.
Thanks, Krisztina. Yeah, that's fascinating. We do have a couple questions in the chat. First one is from Nancy Love. Looks like she revised her comment. So with more nitrogen, more Dauer larva growth, and possibly more oxygen consumption due to microbial activity that creates anaerobic conditions conducive to the larval growth.
Yeah, I agree. I didn't catch the question in there. Was that a question, Nancy, or a comment? Because I do agree.
I had two questions. One, I'm trying to remember, did your C to N ratio change between with and without biochar?
No.
It was the same. Okay. So your total N was the same in both.
Yeah, it was only changing with and without urine, which makes sense, of course. But not with and without.
Yeah, that's what I meant. I mean, with the urine. So I'm just trying to think about things that happen when you add nitrogen that would lead then to more growth of things that would reduce your oxygen, and just thinking about it as what the controls are for the comparison. But I'll chat with you separately. Thank you. That clarifies.
Yeah, I'd be interested to chat.
Yeah. Another question. What is the method for counting nematodes?
Yeah, I didn't go into detail on that. Thank you for the question. So in this case, we were looking at them through a microscope, and we have a method where after doing the extraction and condensing and concentrating the nematodes in water, they fit into a 2 ml tube, and that tube is emptied into a petri dish that we then look at under the microscope. And instead of counting every single nematode, which is often very time consuming and sometimes impossible in the case of Dauer larvae, we have a method where we take 2 diagonal counts and then multiply that by the number of cells, like the petri dish is divided into cells, to get a total number.
Great, thank you. Let's see, looks like it's 12 o'clock, so we need to move on to our next presentation. There are a couple more questions in the chat. Maybe Krisztina, you could offer some responses there.
For sure. Thank you.
Right. Okay, so our next presenter is Trisha Nickerson, presenting on the potential of source separation to improve humanure-based soil amendments for mine closure and remediation. And Trisha Nickerson is a fifth-year PhD candidate in the chemical engineering department at the University of Colorado in Boulder. Wandering is her favorite pastime. She often thinks in metaphors. She is satisfied by simple systems that actually work. So Trisha, you are on.
Thank you. I don't see the ability to share my screen. It says like host disabled.
Okay, give us 1 second.
Okay, sorry.
Oh,
Okay,
you should be able to share screen now. Sorry about that.
Okay. Okay, amazing.
okay, we can see your screen. And so
Okay,
you're up.
amazing. Okay. Um, yeah, so thank you for the opportunity to present today. I'm going to be talking about potentially using humanure on specifically mining impacted land. Let's see. Where my clicker? So this project kind of came out of the thought that oftentimes communities that have underdeveloped sanitation also exist near sources of contamination like mining or industrial sites or landfills. And often they require the soil is lacking the same things that degraded agricultural land would lack. They're usually low organic matter and nutrients and water content, but then also have like additional factors like being acidic pH or having high metal content or other contaminants. And so the idea is, yeah, similar to kind of what everyone else has been talking about, but that the humanure and other organic matter waste streams can be used to help with the remediation of these types of lands as well. And then this is a slide, I'm not going to go through all of it, but just to say that contamination is not just a problem in these kind of like lower income regions, like it's very widespread here in the US and around the world. So there's plenty of space for organic materials. So the question kind of started with like what makes an amendment good for the remediation of specifically for mining impacted soils. And then as I mentioned, one of the major goals of remediation is to increase vegetation to stabilize the soils and prevent erosion and runoff and toxic dust. And then of course to decrease the contaminant exposure so prevent it from being breathed in and getting into water supplies. And so the types of soil properties you would want to promote to encourage these outcomes would be, as I said, to increase the water holding capacity, the organic matter in the soil, nutrients to support vegetation. And then kind of on a chemical level, having attractive interactions between soil particles can help promote soil aggregation to also reduce erosion and give vegetation something to like root down in better. And then in the case of there's multiple different strategies for remediation, but in the case of trying to stabilize heavy metals within the soil itself, the goal would be to reduce the metal solubility and bioavailability. And so this can be done by increasing attractive interactions in the soil to bind the metals. And then in the case of positively charged cations, raising the pH would also be desirable. So the hypothesis is of this project was that kind of a lower tech solution could produce better a better amendment than what are conventional, like co-treated, anaerobically digested biosolids, because biosolids are sometimes used on remediation sites, and that was like shown here. This remediation at the Climax mine in Colorado was completed with biosolids, so it's still, that can be useful, but I guess the idea is that potentially a better amendment would be source separated, aerobically composted human manure, or any other. Obviously, there's a lot of different ways to process it, like biochar also has a place here. So this was kind of just an initial go at it. So the hypothesis was based off of the fact that aerobic compost, is generally more aromatic and oxygenated than digested biosolids. And I was just gonna give like an aside here, just if anyone's not a chemistry person, so this is just a model humic acid, and when I say aromatic, these rings, 6 carbon rings that have this double bonds, create kind of like a delocalized electron cloud that can adopt partial charges and then interact with other aromatic rings. And so this can create those like stabilizing soil interactions that I talked about, and then increasing the oxygen content. So this is a carboxylic acid, and at neutral pH, it loses a hydrogen, and then is able to, and then it adopts a negative charge. And so that provides a binding site for a positive cation like lead. And often multiple carboxyl groups will bind around lead to complex it pretty strongly. So that's the first thing that promotes stabilizing interactions for soil and for metals. And then the other thing is, due to the source separation, just having a lower initial contamination increases the binding capacity for heavy metals. So some past studies kind of supported this idea, just showing, so this study is from Rebecca Riles, and it just shows composted human manure, I believe from soil in Haiti, to biosolids and some other amendments. And really the compost was by far the best at increasing the crop yield over multiple cycles. So the kind of longevity of the amendment is pretty important for revegetation on mining land, just because there's so much of it. So yeah, it's kind of not something you would want to apply many times. And then another study showed that there was a compared composted soil versus soil that just had raw manure added to it. And the compost reduced the amounts of metals that were uptaken into the plant tissues. So this was kind of indicating that compost is reducing the metal bioavailability in the soil. So for my study specifically, and this is kind of actively underway, so I'm just going to share some like preliminary results, but the materials that I collected were from a lead-rich mine tailings pile in Idaho Springs, Colorado. And then I collected 4 different amendments. So the first was topsoil, because this is kind of the status quo for diluting mine tailings after after a mining period. And then I got biosolids from a wastewater treatment plant, composted biosolids from an industrial composter, and then source separated compost from an off-grid community. And then basically just prepared these by drying them, grinding them, sieving them, mixing them. The biosolids I did leave wet, I think because of the polymer addition. They're extremely difficult to dewater, and they would be applied with some level of water anyway. And so I did some initial characterization on the mine tailings and amendments. So kind of as expected, the mine tailings had a low pH, relatively low organic matter content, fairly low field capacity, which is just the proxy for the water retention, and then elevated levels of metals, mostly lead. And then for the amendments, the main thing I'm showing here is just that biosolids had the most nutrients, the nitrogen, phosphorus, and potassium here, higher levels of salts, and higher levels of metals. So that's kind of expected from the co-treatment. What was unexpected was that the source separated compost had a low pH, around 4. So that was kind of unfortunate. As I mentioned, increasing the pH is one of the most important factors for stabilizing lead in soils. However, in literature in the past, I think that this is kind of an anomaly, and that typically source separated aerobic compost has a higher pH than biosolids a lot of the times. So yeah. So anyway, continued with the experiment and basically made these little incubation columns with varying the weight percent of the amendment to the mine tailings. And then maintaining them at a constant water content that was unique to each sample over a 40-day period. And then, yeah, just some initial results. So the next 3 graphs, let me see if I can get my pointer on. On the x-axis have the amendment weight percent, and then these boxes are what amendment it was. So it's topsoil at 20 and 40 weight percent, and biosolids at 5, 10, and 20 weight percent. And then this is just the 0 is the mine tailings alone. And then the water content is based off of a saturation and then a 24-hour drain period, so kind of the field capacity. And basically, the topsoil did not have any impact on the water content, and that's kind of expected. It's not really adding any organic matter. The biosolids had a huge impact on the water content. But you could also ask if you would want the field capacity to be 65%, because that could also lead to waterlogging, I think. So yeah, the source separated compost had kind of more reasonable increases in water content. All of the amendments increased the pH. As I said before, the source separated compost was low pH to begin with, so it did not do that as much. And then the amendments could either increase or decrease the electrical conductivity. And this was kind of a proxy for the binding capacity. So the most obvious one is the biosolids dramatically increase the conductivity. And so you can imagine that if the biosolids become saturated, all of the binding sites become saturated with ions, either salts or nutrients or metals, then they'll dissolve into solution and then contribute to the conductivity of the solution. Whereas with the composted biosolids, that kind of had the lowest initial level of all of those things, which, yeah, we can talk about why that would be. But basically you see a decrease in conductivity, like indicating that it does have the capacity to sorb the metals from the tailings. And then the source separated compost was kind of an intermediate. We saw like decrease at first, but then at the higher loading, an increase in conductivity. So basically, just the next steps for this project is to go a little more in depth on the organic matter characterization. As I mentioned, the composting increases aromaticity and oxygen content, but to kind of look at it for my specific amendments. And then looking at the state of the lead in all of these samples using X-ray absorption spectroscopy is kind of a next step. the soil formation, I'll probe just doing some wet aggregate stability tests, and then I'll be doing a desorption study to look at the leaching potential from the samples. And then I just wanted to end, I guess, by saying I don't mean to like oversimplify the issue, because remediation and contamination is very complex and every site is very unique. So it definitely requires being strategic, and the yeah, so there's just some considerations that I wanted to like point out, that depending on the contaminants, for example, I talked mostly about lead, and which is pretty relevant to cations. But some metals, such as arsenic, typically exist as anions in the environment. So you can imagine that something that stabilizes lead could actually mobilize arsenic. Also, a lot of these contaminated sites have commingled contamination. The concentration matters, because that determines whether or not the amendment has, sufficient binding capacity for that concentration. And then there's also thoughts about that some of the same properties that make compost or other organic matter good for sorbing, complexing with metals, also make them hydrophilic and more soluble. And so you can imagine that if you like apply a bunch of an organic amendment and then got a heavy rain, that you could potentially mobilize more metals by binding them with soluble organic matter. Um, and then obviously there's the quality control issue. So like aerobic compost and source separation are not as regulated. So if you had applied the compost that I got in a large amount, might not have gotten the most desirable outcome. So those are just some things, but I think overall that the point stands that there is a lot of mining and industrial impacted land that could benefit from humanure and organic waste derived amendments, and that in general, the source separation and dry sanitation can produce amendments with less contamination and more flexibility with treatment to produce amendments that contribute to specific remediation goals. With that, this is just, I'd like to acknowledge my advisors and at University of Colorado for letting me work on this project, and yeah, everyone who supplied me materials. Yeah, if anyone has questions or feedback, I would be very happy to take that. Thank you.
Thank you, Trisha. Yeah, that's wonderful. Thanks for sharing all that with us. Let's see. Looks like we have one question in the chat that I'm seeing. Are there concerns about application of biosolids adding additional contaminants like metals and PFAS to remediation sites?
Definitely. I think that there's the thought, like maybe different thoughts, that some people are like, oh, well, it's already contaminated, so it's like gonna improve it, and not that big of a deal. And then I think other people are thinking, well, it's already not so good, and we don't want to make it worse. And I think that just the fact that having the higher contaminant load also makes it a little less effective for the purpose. But yeah, I do think that is a concern.
Mhm. Any other questions from anyone out there? We've got about 1 minute until we're gonna go to break.
More of just a suggestion, if you haven't connected with Kevin Orner already from West Virginia University, he's been interested in researching this intersection of, I think, acid mine drainage remediation and human waste reclamation. So he might be an interesting person to talk to if you haven't already been in touch.
Yeah, thank you so much. I definitely will look at that.
Yeah.
Yet another connection here at the summit at the Rich Earth Institute. So it looks like it's time for a break. We're going to take a 10-minute break and reconvene at 12:30.